All right, let's get right into it. Today, we are talking about what's called the last mile of computational biology. It's this idea of taking a digital protein that's nearly perfect and making it truly perfect. It's the final and maybe most critical step in a massive revolution, and we're going to see exactly how scientists are closing that gap.
So we all know the story by now, right? AlphaFold2, or AF2, was just this seismic shift in biology. This incredible deep learning tool came out of nowhere and started predicting the 3D structure of proteins with an accuracy that people thought was decades away. It was, and still is, a total game changer.
And that really gets us to the heart of the matter today. An AlphaFold2 prediction is an amazing starting point, but it's not always the finish line. You see, there's a really subtle but super important difference between a structure that looks right and one that's actually ready for serious, high-stakes science. So, what's that missing piece?
Well, that missing piece is what researchers call the last mile problem. It's that final, often painstaking process of refinement that takes a brilliant prediction and turns it into a scientific tool you can actually rely on. So, what are these errors we're talking about? Think of it this way. AlphaFold2 gives you this beautifully assembled car, right? But maybe a few bolts aren't tightened on the way, or a wire is just a little bit out of place. For a protein, that means atoms are bumping into each other when they shouldn't be, or the bonds are at weird, physically unlikely angles. Now, these flaws are tiny, but they can throw everything off if you're trying to do something precise, like design a new drug. Now, for years, scientists have had a way of dealing with these kinds of issues, a way to basically let the digital molecule kind of relax into a more natural, stable state. But, as you can probably guess, that old-school approach came with its own set of serious problems. The classic tool here is called molecular dynamics, or just MD. And what you do is you essentially put your digital protein into a simulated box of water, and you let the laws of physics just take over. The idea is that all the little atoms will wiggle and jiggle their way into a more stable, lower-energy shape, which, in theory, should be a more accurate one. It's a powerful concept, but it's not exactly foolproof.
And this quote from the source paper just says it all. Here's the catch. When you just let an MD simulation run completely unrestrained, the structure can actually get worse. The physics simulators we have, they're not perfect, and so the model can literally drift further and further away from its correct biological shape instead of getting closer to it.
So you can really see the difference here. It's all about control. An unrestrained simulation is a total gamble. It might just wander off in completely the wrong direction. What science really needed was a way to hold on to the good parts of the structure, keep them locked in place while only letting the flawed parts move around and fix themselves.
And this is where we get our first huge breakthrough in modern refinement. It's a method called ReFOLD3, and it introduced this really clever new strategy of using gradual restraints to solve that whole drifting problem.
The ReFOLD3 pipeline it's so smart. It's basically a four-step process. First, it assesses the model. It uses a tool to score every single little piece to figure out which parts are rock solid and which are kind of shaky. Second, it restrains it. It applies these strong digital clamps to the high-quality parts to freeze them, while using much weaker clamps on the flawed areas, giving them room to move. Third, it cross-checks the model against other data to make sure any changes are actually biologically correct. And finally, it runs the refinement, the MD simulation. But now, it's not a wild ride. It's a super-targeted, gentle correction focused only on those shaky, low-confidence spots.
So, did it work? Oh yeah. At CASP 14, which is basically the Olympics of protein structure prediction, ReFOLD3 ranked in the top 10 for refinement overall. And what's even more impressive, it was a top 5 method for refining models that were already really good, proving it could handle that crucial last mile with amazing precision.
But the story doesn't stop there. The next big leap forward was honestly even more elegant. It was this beautiful and kind of ingenious hack that turned AlphaFold2 into its own best refinement tool.
Just think about that for a second. What if the very same tool that created the nearly perfect model was also the best tool to fix its own tiny little mistakes? I mean, that was the brilliant, simple insight that led to this whole AlphaFold2 recycling idea. And the change is almost deceptively simple. I mean, look, normally you give AlphaFold2 a protein sequence, and it spits out a structure. Easy. But with AF2 recycling, you give it the sequence plus a starting structure, the very model you want to fix, as a custom template.
So how does that even work? Well, AlphaFold2 has this built-in feature where it internally refines its own predictions over and over again. By giving it your own model as a starting point,
you're essentially hijacking that powerful internal system. You're telling AlphaFold2, hey, don't start from scratch. Start from here, and use your incredible brain to make this even better. It becomes this superpowered refinement loop.
And the results? They are just stunning. For certain kinds of models, this recycling hack achieved get this, a 100% improvement rate. Every single model they tested got better. That's just an unbelievable level of consistency.
And yeah, this was no fluke. As you can see here, the AF2 recycling method just consistently makes pretty much every kind of protein model better. Doesn't matter if it's a single protein or a big complex, or even if it was made with AF2 in the first place. The success rates are always way up there, in the high 80s and 90s.
Okay, so this is all really cool from a technical perspective, but let's take a step back. Why does any of this matter so much? Why are we spending all this brain power to fix these tiny atomic-level flaws?
It matters because closing this last mile gap is what takes a model from being almost right to being ready for the lab. It's what unlocks our ability to do high-accuracy drug discovery. It allows us to do precise protein engineering, to really get a deep understanding of how diseases work at a molecular level. It basically brings these computational models up to a standard that can rival actual time-consuming experiments.
So, the bottom line is this. Methods like ReFOLD4 and AF2 recycling give scientists confidence. They give them the tools to not just find the tiny flaws in a model, but to systematically fix them. And that transforms a theoretical model into a reliable, trustworthy tool for making real-world discoveries. And that really brings us to our final thought. Now that we can reliably create and perfect these digital proteins, getting them to what we call experimental quality, what's next? What new, even bigger biological mysteries are we going to be able to solve when we can simulate life with this kind of accuracy? The last mile might be closing, but a whole new frontier is just opening up.